Hello everyone and welcome to my talk. My name is Jonathan Sheavly and I'm a fourth year graduate student at the University of Wisconsin-Madison, and I'm going to be discussing the simulations of the bilayer mediated assembly of cationic nanoparticles on phase-separated lipid bilayers. Nanoparticles are particularly special because they have a large degree of tunable size and surface chemistry. Over here what we show is that experimentalists have the capabilities to create nanoparticles at whatever size is desired at a very narrow distribution. What we also see is that the experimentalists can also covalently conjugate different functional groups onto these nanoparticle surfaces, providing a large degree of tunable surface chemistry. And these gold nanoparticle properties are designed for a very specific application in mind. Specifically, cationic nanoparticles, which we're interested here, have a variety of potential biomedical applications. And these typically require assembly to the bilayer. So for this first example, we have phototherapy, which is where we have a mouse with a tumor, and we have a collection of nanoparticles that we inject within the mouse. Those nanoparticles will deposit selectively on the tumor, and once they deposit on the tumor, you can have a laser shine on that deposit. Now what's special about these deposits is that they have increased photothermal efficiency from isolated nanoparticles, allowing you to increase the efficiency of heating those cells and destroying those cells in comparison to other cells. So this provides a selective method for treating these types of cancers. These can also be used for selective diagnostics. So in this specific application, the mouse here will be injected with nanoparticles, which you'll see in this injected site, but you will also see that the nanoparticles selectively adsorb to the tumor sites and not anywhere else in the mouse. So this means that where you see the aggregation of nanoparticles on these tumors, these are the places where the tumors are predicted to be, and you can diagnose, make a cancer diagnostic with this tool. So the key question that we want to understand is how can cationic nanoparticles aggregate for these applications, since we all know that cationic nanoparticles are intrinsically repulsive of each other. So to understand how materials interact with the lipid bilayer, we wanted to first investigate the role of curvature of these adsorbates. And in nature, this occurs with peripheral membrane proteins. And specifically with this one, it's called the bar protein, which absorbs onto the surface here. The free energy is actually minimized when they're near each other rather than when they're far apart from each other, because the curvature is minimized in this case. And when you scale this calculation up to the size of a vesicle, what you see is large-scale self-assembly and reconstruction and remodeling of the lipid bilayer. In our previous work, we started to look at how nanomaterials interact with specific bilayers depending on their composition, whether or not they were a liquid disorder bilayer, which is where they're a little bit more flexible, or liquid order bilayer, which is where they're more stiff. What we found is that when nanoparticles absorb to the liquid disorder side of the boundary, you get many contacts, as you see in the liquid disorder case, but you have less induced curvature since the curvature is built into the bilayer, and therefore we saw increased partitioning of the nanoparticles at these phase boundaries. And furthermore, what we saw from our collaborators, which published a paper this year, found that nanoparticles bind near the RAF boundaries, suggesting that our predictions were correct in the atomic force microscopy measurements. So this bogs a key question. Can curvature actually drive assembly of nanoparticles in real systems, just as it drives partitioning to the RAF boundaries? And the other thing that we're thinking about is we're going to use MD simulations to find out. So our MD simulations are set up using a coarse-grained simulation. We use a Martini representation because this is very widespread and a very common lipid bilayer construction. We're using the DIPC lipid for the liquid disordered case, and we're using the simple cationic ligand for the nanoparticles. In this case, we're also using implicit solvents, so we can scale up these simulations much larger. And then we're going to each model the one-phase bilayer interactions and the solution, and then we're going to move on to the two-phase bilayer interactions. So first, our unbiased simulations with multiple nanoparticles, we're going to be using a clustering algorithm for a system of 20 nanoparticles. So we can measure the degree to which nanoparticles associate in different environments. And we compare just nanoparticles in solution, nanoparticles bound to a liquid disordered bilayer, and nanoparticles bound to liquid ordered bilayers. So we can run these unbiased simulations, and visually you can see that the liquid disordered bilayer cluster much more than the liquid ordered bilayer, which more than the in solution. And we can quantify that using a cluster size distribution, where we measure the probability as a function of the actual cluster size. So what we see is that nanoparticles don't aggregate ever in solution. They do form many clusters in the liquid disordered absorbed state, where you see this large widened distribution of cluster sizes, and they do form a few clusters near liquid ordered bilayers. So this suggests that there are some bilayer mediated interactions between adsorbed nanoparticles, which are particularly strong in this liquid disordered case. And this is consistent with some experimental observations that we've seen earlier in 2016, where we saw nanoparticles associating with each other when they're absorbed to a bilayer. So in order to understand these interactions a little bit more clearly, we decided to measure the free energy profile and directly probe the two-body interactions. So we perform umbrella sampling to measure the free energy profile for associating nanoparticles. So we have here the potential mean force in kilojoules per mole, which is the free energy projected onto one reaction coordinate, versus our nanoparticle distance as measured as we see here in this schematic. And the key findings from this are that nanoparticle association is favorable in the LD bilayer, where we see a global minimum here in the associated state. We see slightly unfavorable in this liquid ordered bilayer, as was shown here, with a positive free energy change, and then unfavorable in solution, where we show a 14.6 kilojoule per mole free energy local minimum here, which suggests that that is thermodynamically unfavorable. So this is consistent with our cluster size distributions that say that only the liquid disordered case showed a large degree of clustering. Now, for the liquid disordered case, which is the case that showed the most favorability, we wanted to decompose the free energy profile into components to understand the nature of the interaction. So the difference between, we broke this down first into the delta G bilayer, which is the difference between the liquid disordered and the solution potential of mean force, shown in this black line. We also measured the curvature free energy, shown in this red line here, and the direct interactions between the nanoparticles and the lipid bilayer, shown by delta E LJ elect, which is in blue here. So what we see from this plot right here is that our long-range interactions, we see a decrease in the curvature free energy that corresponds with the difference in bilayer free energy, suggests that those long-range interactions, so above this 6.4 nanometers, are mediated by curvature. However, after we go past that 6.4 nanometer distance, what we see is this delta G bilayer tends to follow the Lennard-Jones plus electrostatic interactions, suggesting that those short-rate interactions are mediated by direct interactions with the bilayer. Now, we wanted to understand and extend this understanding to three-body interactions. This is where we have the attraction between three different nanoparticles on the bilayer. So this is because the liquid disordered bilayers in the clustering analysis demonstrated that clusters of 3 nanoparticles were possible. And so therefore we measured these three-body interactions as well. So we compared the solution case with the LD adsorbed case in this figure here, where we had the same potential mean force versus the nanoparticle distance. And what we see is we show a positive free energy change for the liquid disordered state, but it is still lower than the in solution case. So the overall conclusion from this is that trimerization is less favorable than dimerization. Then, given that trimers can occur in the clustering analysis, and they may be unfavorable in comparison to the two-body interactions, we'd like to see if they form lines or prefer close-packed aggregates. So what we see here is a potential mean force versus the 3 nanoparticle angle as demonstrated by this convention shown here in degrees. And what we see is for both of these a local minimum in the 120 degrees, which we call the close packed state, and a global minimum at the 0 degrees. And for this, what this means is that trimers, when formed, do perform linear aggregates, which is the 0 degree case. However, it does seem to show that the LD adsorbed case seems to stabilize the close packed state. The curvature free energy difference is not enough for this interaction to be favorable in order to form close packed. So finally, in the last part of this, we wanted to measure assembly near phase boundaries. And so we can model these phase boundaries as we've shown in our previous work, and we can throw a bunch of nanoparticles on those phase boundaries. What we also notice is we do see these accumulations near those near those phase boundaries, so we'd like to look at the energetics of that association. So first, what we do is we measure the free energy profile as a function of the nanoparticle distance, while both of them are constrained to be near the boundary. And what we see is a free energy profile that looks very similar to the bulk liquid disordered bilayer. So this means that this association would be just as favorable as you would see in the liquid disordered bilayer, which is exactly what we saw was favorable in the liquid disorder case. Moreover, we decided to measure the potential mean force versus the angle with respect to the RAF boundary to see if it aligns parallel or perpendicular to the RAF boundary. So based on this sign convention here, what we show is that the 90 degree case, which is where the nanoparticles are aligned along the RAF boundary, is the most favorable case, and there's a large barrier for reaching the metastable orthogonally bound case. So the conclusion from this is that alignment to the boundary is more favorable than disalignment perpendicular to the boundary. So in conclusion, what we'd like to say is that bilayer curvature can drive association of adsorbed nanoparticles. For the given size and surface chemistry, dimers are the most favored on the liquid disordered bilayers. Longer clusters are unfavorable, but when they do form, they'll tend to form linear aggregates as opposed to close-packed aggregates. Dimerization on the RAF boundary is also favorable, very similar to the liquid disordered case. And then what we also found by doing the angle analysis is that dimers will align along the contour of the RAF boundary. So with that, I'd like to acknowledge and thank some of my group members, my advisor, specifically Reed Van Lane, shown here, and also their postdoc, and our 3 older graduate students who will have presentations here at AICHE as well. So I invite you to go check out their presentations. I'll leave links on a part of my poster for their talks. I invite you to go look at them. I'd also like to thank my funding sources as well and acknowledge them. And so with that, I'd like to thank you for listening to my presentation and taking the time to learn a little bit more about these nanoparticle systems. I would invite you to check out this paper down here, which is where this work was published, if you are interested in learning more. I also invite you during the allotted chat time to meet me on my posted Zoom link, which I'll put on my poster. And with that, I'd be happy to take any questions, and thank you very much for joining my session.